Welcome back to our second installment on our series on epigenetics. Today we dig a little deeper and take our inspiration from the original work that Michael Meany had done at McGill in Canada. This is now an extension of this work done by the group at San Diego, the Salk Institute, run by Fred Gage, who has done a great deal of innovative work in neurobiology. Briefly, he believes that life experience shapes the neural genome, and here's an excerpt from a brief discussion he had with a magazine analyzing this paper that had appeared in Science. So they transposed the work done on the glucocorticoid receptor modulation by epigenetics to a phenomenon of transposon integration into cells in the rat hippocampus. And as it turns out, that the quality of mothering, just as it did in the glucocorticoid receptor story, also appears to play an essential role in the liability of transposons to be activated in the rat genome in the hippocampus and be integrated into various locations in neurons in a mosaic type pattern. So not all cells would be liable to have transposons be integrated, but there's a mosaic type random selection where this transposition occurs and perhaps impacting the function of these cells in some kind of major negative or perhaps a positively creative way, increasing the diversity of neuronal function in the hippocampus. So this was quite a mouthful of an introduction. Let's step back a little bit and look at the background. So we have known for a while now that different upbringings will affect your epigenome in different ways. So here you see the attentive mom taking care of the rats here, and here on the other hand you can observe a neglectful mom that maybe she's depressed, not attending to her pups very well at all. And here's an allusion to the socioeconomic background, not perhaps of these rats, but of humans. Here you see a neighborhood in a more higher class, high socioeconomic stratum, and here's more of a slum-like, lower class, tenement type neighborhood, and these differences also have been shown to play a role in the epigenome. Here then is the Michael Meany story in a brief summary again. You can see here the quality of the mothering is thought to leave epigenetic marks, in particular the methylation of cytosine residue, leading to differences in gene expression and then into differences in the inherited phenotype in the mouse circuitry. In this particular case, he was focused on the glucocorticoid feedback. As you can see here on the left-hand side, poor grooming leads to a decrease in the glucocorticoid receptor expression, which then makes the system much less susceptible to feedback by cortisol, inviting runaway stress hormone escalation in the system. Here is an attentive mom who expresses appropriate levels of the glucocorticoid receptor, tuning the system to be very responsive to circulating levels of cortisol. Here is the total outline. Here's the hippocampus, hypothalamus, corticotrophin releasing factor, of course, plays a role, which goes to the pituitary gland, and eventually ACTH stimulates the adrenal gland, and in a feedback fashion the glucocorticoids can cycle back to the hippocampus, bind to the glucocorticoid receptor, and shut this increase in production down, at least in a temporary way. So So that is the first piece of information we need to know to understand the paper produced by the group surrounding Fred Gage. Here is the other piece, and it goes back to Barbara McClintock, who started at the Cold Spring Harbor Laboratory by studying maize, because she was struck by the fact that maize has these different shades and colorations, often within the same kernel, and the question is, how is this possible? And she proposed early on that she suspected jumping genes. Now, she was ridiculed for this, in part perhaps because she was one of the early women scientists in the field, and her idea was so explosive and innovative that she met with a great deal of resistance. However, she was awarded the Nobel Prize eventually for this work. So we have the idea of jumping genes, or mobile elements in the genome that, given the right conditions, can be activated, transcribed, and reintegrated at random sites in the genome, creating these kinds of mosaic expressions, in this case of pigments. Now, we all have a lot of transposons in our genome, and it appears that there is a progression in how many of these mobile elements we have in our genome according to our evolutionary status. So here are the invertebrates, here are the chordates, here is our friend the dog or vertebrates, and here are we. So you can see that the amount of transposable elements in the genome has been increased through the development of the mammalian line. This gives you a breakdown of the total contribution. So perhaps only 2% or so of our DNA are true coding sequences encoding our proteins in our own cells, and the rest are all kinds of non-coding sequences, in part transposable elements, which we will address now in this talk as being important perhaps in the epigenetic modulation of neuronal activity in the hippocampus. So a brief review of the epigenetic phenomenon. You can see here that the chromosome is very tightly packed. Chromatin is a bioengineering feat of packaging DNA in association with histone proteins. Here is the histone. You can see that histone tails can be modified by acetylation or by addition of a methyl group. In addition, DNA itself can be modulated at cytosine residues, particularly in those areas which are GC-rich islands that lend themselves to methylation. And these two events are the major modifying events, modifying the gene expression without modifying the DNA sequence. So here is another way of showing this. You have here the polymerase, RNA polymerase, which can bind with certain transcription factors to a gene and then lead to a transcript, which can then be edited and either produced into a protein or reintegrated into the DNA in case of a retroposon. So here then is the story that Fred Gage's group was dealing with. He believes that maternal care can alter genomic structure, and the mechanism of this will be to activate latent transposons, especially of the L1 type, because the methylation of certain receptors and promoter sites that promote the production of these transposons might be deficient in rats exposed to poor mothering. And this is exactly what would then lead to the phenomenon of mosaic cells, namely cells that have certain genes expressed neighboring cells, different genes expressed, both resulting from the same progenitor cell. So the progenitor cell is then modified by the random transposition of these line 1 repetitive ancient elements into new DNA targets, perhaps enhancing or minimizing or even destroying the activity of certain genes. Here you have this typical line 1 element, and it is read or transcribed from the 3' end backwards. So the group in San Diego developed assays where they could detect in the cells in the hippocampus transcripts of increasing lengths, starting here at the 3' end and working their way backwards. Here is another slide that shows this. The L1 elements mobilize through a copy and paste mechanism in which full-length L1 messenger RNA is reverse transcribed at the beginning of the 3' end and inserted into new genome locations. So sometimes reverse transcription stops early and immediate single-strand DNA molecules are degraded because insertion can be completed. So the assays that they worked out then are able to detect progressive transcripts starting from the 3' end and perhaps in successful cases going all the way back to the 5' end, allowing for a complete insertion of a completed transcript of an L1 transposable element to be inserted into the genome. So here then is what it looks like in the brain. You have progenitor cells, neuronal progenitor cells, in which this particular transposition event takes place. So here you have the red cells where the transposition has occurred, and then these will be incorporated into the circuitry, resulting in a mosaic assembly of cells that either have this genetic modification or not, being derived both from the same neuronal progenitor cell. Here is a more detailed presentation of this. You see the line 1 element here, and you see the different methyl groups attached, suppressing the transcription of this line 1 element at the promoter sites. However, under certain circumstances, this can be relieved. The suppression by methyl groups can be relieved, and you get a transcription of the entire open reading frame and insertion into a new genomic site. So here then is the result of the experiment. You can see that maternal attention to the offspring follows a certain distribution. Not all mothers are terribly good and not all mothers are terribly bad. It's kind of like a Gaussian distribution. And what the group did was divide the mothers and their pups into two basic groups, namely the high maternal care and the low maternal care groups. And you can see here broken down the different maternal behaviors that can be looked at postnatal day 1 through 14. So this is the critical period that the researchers looked at. And you can see that here in the high maternal group, you have a great deal of licking, grooming here, exceeding that of the low maternal care group. So these are the measures that were used to divide the two moms and their pups. Cross-fostering experiments were conducted as well, just like as Dr. Meany did at McGill. So here is the result. This slide says it all. So here you have a measure of the transcripts. How many copies of this line 1 element were transcribed in these cells in the pups, according to low mothering and high mothering kinds of scores. So the best mothers had a score of 100, the worst mothers had a score of 85 or so. And you can see that poor mothering is associated with an increased availability, increased transcription of line 1 elements in these cells. And you can see the different transcripts. This is from 3' end, 5', and here they completed all the way to the 5' transcription. But all transcripts correlate negatively with the degree of mothering. You can see that not all transcripts make it to the entire completion, but you can say the attempt of the transcription machinery, the unleashing of line 1 transcription is proceeding. In the next slide, you see another control experiment, namely extracting single nuclei from these cells by cell sorting, fluorescent cell sorting, which allows you to separate cells which have been tagged with a label from other cells, and using a very sensitive PCR, polymerase chain reaction assay, to look for L1 copy number. So this is an amazing feat of micro-analysis by looking at single cell level PCR, and you can see here again that high maternal care is associated with lower transcripts compared to low maternal care. And this is particularly true in those cells, those cell nuclei that have been isolated. The effect gets enhanced at the nuclear level because that is the site where DNA, RNA transcription, and then DNA reintegration takes place. Finally, the group then looked at the L1 promoter, that site which invites transcription factors to come in and turn on the transcription machinery, and looked at the methylation state of this promoter site. Again, taking the inspiration from the work by Dr. Mini, who also had looked at methylation at the glucocorticoid receptor promoter site. So here we're looking at the L1 promoter. And this promoter contains a very large number of repeat monomers with each representing a GC island, inviting literally the methylation to come in. And you can see here that this is an active promoter which behaves in the appropriate fashion. Now, when rats were analyzed according to high mothering and low mothering status, the pups in other words, and you look at the methylation intensity at this particular promoter site, it's called YY1 promoter site, you can see that methylation is much more pronounced in the high maternal care group. There's more red color here than blue color if you compare the intensity of the bluish areas and the red areas. So that would then confirm the prediction that the mechanistic aspect of how poor mothering is translated into a change in line 1 production and reintegration is controlled here by methylation or the lack of methylation at the appropriate promoter that leads to L1 transcription from the genome. And here you can see on the right hand side that maternal care again correlates with the percent of methylation of the receptor. Methylation leading to a reduction of the promoter, shutting down the transcription of the L1 element, thereby leading to less RNA production, reverse transcription, and reintegration into the genome of the L1 transposon at various sites that are somewhat random, leading to mosaics in the brain, affecting the function of these mosaic cells in a somewhat unpredictable and as yet unknown fashion. So the analysis then here needs to really go down to the individual cell level to demonstrate what is the impact on the wiring pattern of these brain areas as impacted by the deep reach of environmental events deep into the genome of brain cells, rearranging, in this case, the genome itself, not just a biochemical effect such as a modulation of a receptor, but rather a pronounced effect on genome reshuffling, which is heritable throughout the progenitor cells that build up the neural circuitry. Now, lastly, just an advisory in the last slide here. It was recently reported that childhood stress and adversity also can result in hypomethylation of retroposons in humans. By retroposons in humans is meant, of course, the chromatocytes that allow for the production of retroposons. So that was it for a report on how genome can be reshuffled by deep environmental grip into the genome itself through epigenetic mechanisms, and how this can lead to mosaic expression of different gene patterns in the genome of rats. We will continue this series soon with a report on how trauma has been found to influence the genome through epigenetic mechanisms. In the meantime, we thank you for your attention. We'll see you soon again for a talk at Behavioral Health 2000.